Right. Good evening, everyone, and thank you for joining today for the Institute Colloquium. It's always good to see a full house. Those who are waiting in the wings, please do come in quickly. And sorry about the slight delay in starting. So we have amongst us today a scientific luminary who has pushed the boundaries of our knowledge regarding sort of the genetic and molecular language used by nature to sort of construct one of the most complex structures we know, which is the human brain. Okay. So Professor Shubha Tole from TFR Mumbai is a world-renowned neuroscientist who has done some groundbreaking research that has sort of illuminated or thrown light on the mechanism on the development of the mammalian brain. So her work addresses the interesting question about how a collection of cells without particular form or shape sort of develop into something as complicated as our own brains. So she's internationally recognized for her role in discovering various mechanisms by which this complicated process happens, and I don't claim to know anything about it. So that's why we are all here to learn about that. And of course, one can easily see the implications or the importance of the work, because it certainly throws light on how various neurodevelopment disorders and problems come about. Because if you understand the process, then you can also start understanding what goes wrong and then try to fix it as best as possible. So for her pioneering work, Professor Tole has received several awards and recognition, including the Shanti Swarup Bhatnagar Award. She's a fellow of all three science academies. She's also awarded the Wellcome Trust Senior Fellowship in the recent past. And she is also known for various other roles, including as a science popularizer, public outreach with media engagement, being in various advisory roles and sort of championing gender equality and various other roles. So without further ado, let us hear Professor Tole talk about the topic, the circuits of sensation.
Slides and pointer. Yeah. Okay. Thanks so much. Thank you so very much for inviting me and hosting me so warmly. I've had really nice interactions with many people, a few students in the labs, some faculty members over lunch with your directors, deans, and so on. I look forward to sharing some glimpses of the excitement that I have been privileged to enjoy because of the wonderful students and postdocs who have opened amazing new windows into how this fantastic computer develops. So I'm a neuroscientist, that means I study the brain. I'm a developmental neuroscientist, which means I study how this fantastic computer is assembled. It's not a structure, it's not a machine or a computer that can be assembled by buying parts. Each part has to be grown in the right place and then has to be wired up. So it's a biological problem where the hardware gives rise to the software. So the equivalent of loading a program onto your circuits is essentially modifying the circuitry, strengthening synapses. So hopefully by the end of this talk, some of your synapses will have strengthened, some may have changed. So the software is that. So essentially if the hardware does not grow and the circuits don't connect, function will not come. Our brain functions the way it does. Our circuits enable certain functions and they do not allow certain other functions, okay, just because of the way they are constructed. I study, one whole direction of study in my lab is how these circuits are actually grown. And say something that sounds a little too complicated, just raise your hand and I will make it simpler, because I think there is no science that cannot be communicated to a wide audience. So here we go, the circuits of sensation. This is the human brain circuitry. Okay, it's from the Human Connectome project. This is the most complex brain we know. This is not a model system for anything, but it is what we seek to study using simpler model systems. And in my lab, we use the mouse, which is a mammal. And to the extent that we understand the brain, the mouse and the human are very, very similar to the extent that we understand how it develops. The same mechanisms play out. Here, the imaging program that is used is such that all of the wires in the brain are labeled, and every time they change direction, they take on a new color. So this entire thick sort of cable, okay, it's like that flat computer cable. It is actually the one that connects the two halves of the brain, and it turns in and here. So every time it changes direction, there's a new color. And we seek to simplify this problem by studying individual circuits. Here is a section through the brain, and you can see the beautiful forest-like neurons, okay? And again, it's thick with neurons, and I'm going to share some data about some of the wiring circuits that creates the wiring of the cerebral cortex, okay? What does the brain do, okay? Gerard had this very interesting statement that illusion is in fact the perceptual rule, not the exception. What can that possibly mean? Okay, and I'm going to show many examples that bring home this point, that the world is out there, it comes in through our five senses, and yet what our five senses present to the brain and what we perceive from it is one version of reality, if one could even define what reality is. Okay, for example, I'm sure we all see, I certainly do, that this is a nice white piece of cardboard, chart paper maybe, that is a nice square that is placed on top of 4 black discs. And in fact, if you look carefully, you'll even be able to see that the white of the square is perhaps a little creamier than the white of the background, which is a little grayish, right? At least I see that. How many agree? Yeah, that's what our circuitry is telling us, right? And okay, if you all don't see it, come and see me outside. I'm a neuroscientist. Okay, and yet this is actually one of many types of illusions. It's a Kanitsa illusion, where all I have done now is place more squares on top of this, and you see that the line disappears. And now there is no creamy versus gray line. What this was an illusory contour. Okay, the line there was a complete illusion created because our brain is wired to see complete shapes. It completes shapes for us. Where boundaries don't exist, it adds boundaries. And this is vital for our survival. Okay, this is not just a funny party game. This is vital for our survival. Okay, so vital that even animals need it. Okay, we all know kittens will sort of cram themselves into any sort of bounded shape they see. Okay, even when they don't exactly fit, they will make themselves fit. There was a lovely study modeling kittens as liquids because they can just flow from one liquid takes cell. Going back to the Kanitsa illusion, scientist Gabriela Smith conceived this lovely experiment in the middle of the pandemic when nobody could actually go and do any experiments. She sent kits to, not kittens, but kits, to various people in New York, cat owners, and essentially asked them to make squares on their floors and count how often a cat sits inside them. Okay, she called it, if I fit, I sit. And then she did this experiment. Okay, she basically gave this kit and asked to count whether a cat would preferentially sit in a Kanitsa illusion square versus not. Yeah? And the data really was biased towards this, that even a cat can see these illusory contours and will more likely sit in an illusory square than not. And this is important for survival. I keep saying this, and you all are kind enough to say, okay, where the hell is she going with this? But essentially, image completion is what we do. Okay, this, all of you will tell me, is a Bisleri bottle. Okay, and that is not the most minimal description of this. The most minimal description of this would be, there's a bit of plastic here, and then a bar, and then another bit of plastic, and then a bar, and so on. Right? We think we see a Bisleri bottle because, okay, and especially if the object moves, we will suddenly tie the bits of the bottle together and make a moving object in our minds. But what we are seeing is not the full bottle. And indeed, out there in the world, most of the time, we do not see the full object. Imagine if we decided, oh, this is not a full tiger, it's only bits of tiger. Okay. You see why I said this image completion is a survival, necessary for survival? The tiger itself would go hungry if it thought, oh, this wasn't a full deer, it's only like the deer's bum and the deer's neck. I'll wait for a full deer, then I'll bother. Okay, how much of the world do you see today is intact, entire images? Hardly any. Okay, when you go outside the room, you'll see how many things are all just partial images and we complete them, right? Our brains are wired for this. Our brains have circuitry that can extract shape from bits and pieces with Kanitsa illusion contours. And in fact, even entire shapes from just random dots. Have you go to YouTube and see that there are videos of random dots moving around on the screen. And if even 30% of them are correlated, you will suddenly see, you will draw a shape around those that are moving correlated and say that's a ball. Okay, there are experiments that do this. How much correlation do you need in order to extract a shape from what is absolutely programmatically dots? No shape in there, right? So we extract this. Our circuits are evolved for this. And if they don't wire up correctly, we're going to die either hungry or inside somebody's stomach. Survival, okay? So I'm going to spend the first few minutes of the talk motivating how circuitry forms is vital to circuitry function, okay? Because my focus is then how does circuitry form, and then I will show you some data, okay? Here I'm going to show you yet another example of circuitry at work, okay? I'm going to make you see sound. This is called the McGurk effect, and I'm going to see if I can.
Ba, 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 ba.
Okay, how many of you heard toddler ba 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 ba? It's okay, that was all that was playing. There was no hidden sound and all. I hope you all heard ba 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 ba. Nobody? Okay.
Ba, ba,
Now?
ba, 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 ba.
Okay. The first time nobody agreed with me. Now I am a little bit blind because I need my near sight, so I played the wrong video and I wasn't looking at the video. Because I wasn't looking at the video, I heard ba ba ba. You all were all looking at the video and you all heard va va va. It was the same track each time, okay? Here, watch them both.
Ba, 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 ba.
So now you hear with your eyes? Yeah, it's quite amazing. You want to close your eyes and open them and do the trick again? Your circuits will make you. This is not a, there's nobody tricking you. Your circuits will do it. And they need to do it reliably. Okay, so here,
Ba,
enjoy.
ba, 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 ba.
Yeah? Did it work? How many did it work? Yeah, so you are literally modifying your percept of the sound based on what you see, okay? And indeed, we use this in our daily lives, right? How many times you've been in a busy room and there's a conversation going on, and if you can see the person's lips move, you can somehow hear better. Yeah? Yeah. In fact, I am playing right now a monstrous joke on you guys right now, okay? Am I actually the one who is talking? Where is the sound coming from? If you close your eyes, where is the sound coming from? Speakers, there. And if I move my lips in sync with that, you ascribe the sound to me. There could actually be another speaker back there. How do you think ventriloquism works, right? The puppet moves its lips. And you immediately ascribe the sound to whatever is moving, right? So our brain circuits are designed for this, and this allows us to function in the world. Okay? Oh, okay, please, no more Baba. Please. No. Okay. Okay. How do these circuits arise? In order to form the brain, you have to, in order to form circuits, you need brain cells, you need neurons. In order to have neurons, you need to have a primordium of mother cells, stem cells that are going to produce these neurons. You need to start from one cell, the egg. It becomes 2, 4, 8, 16 ball of cells. Hey, how many people in this room who don't have biology after 12th? Oh no, you all are IISER students. You all have to, whether you like it or not. How many of you didn't enjoy biology? It's okay to confess. How many of you grew up with this idea that biologists are weak in math? Okay, you know, I'll tell you why. Watch all those cells divide, right? Okay, sorry, stop. One cell becomes 2, 2 becomes 4, 4 divides and becomes 8, 8 divides and becomes 16. In biology, division and multiplication result in the same thing. Okay, so here, watch the brain grow, okay? What you saw here is lots and lots, so every little brown speckle is approximately the size of a cell. Let's see, where's the pointer? Yeah. Every little speckle is approximately the size of a cell, and what you saw is lots of cells streaming over the outside of a frog embryo, and they're going to go through that little hole and double up on themselves, and then magically a whole sort of flat plate of cells will arise from which the entire nervous system forms. So just watch it happen. That's going to become the tail, and now watch the spinal cord forming and the front of the brain closing up. And here, if you squint, you can see the beginnings of vertebra, and that's the big bulgy brain. Okay, that's a tadpole, but our early development is no different. So a lot of cell movement, and these are all mother cells, they're all dividing. They will eventually produce neurons, which once a neuron is born, that's the neuron for your whole life. Okay, so one wonders why there are disorders of aging, Alzheimer's and so on. Imagine the neuron that is born when you were in your mother's womb. That's the neuron and all its connections as they grew, as they learned or they didn't learn or whatever. That's a neuron that you have when you're 80 years old. Good God, I mean, which piece of hardware lasts that long, right? And there's no replacing it. Yeah. So modern medicine has allowed the rest of our hardware to last longer, or if not, we can get artificial knees, artificial whatever, you know, we can get transplants of bits of liver, we can even get a donor heart, but until now there ain't no donor brain. So the neurons that you were born with are pretty much, barring a tiny few locations of exceptions, no new neurons. So that's the hardware you have to work with. And that is what we're trying to study then. Yeah, so to make the problem a little simpler, no, well, okay, this is the problem statement. The human brain is considered to have 100 billion neurons, and each of them making between 1 and 10,000 connections. So this is a typical neuron in the cerebral cortex, and each red dot is where some other neuron has come and connected to it. Okay, imagine the wiring diagram to wire this up. Okay, no electrical engineer would want to take on this problem, and yet the mouse's embryo does it, the fish embryo, the frog embryo does it reliably. Okay, all right. Watch one neuron grow in a dish. This is a video taken through a microscope by scientist Tara Lindsley. This is the neuron in a dish. Okay, and as it grows, it will send out many little tester sort of wires. They're called neurites. And some of them will be the input wires, but one of them will decide and become an output wire, and it will grow towards its target. So a neuron has to decide, one important cell biological decision is, will the wire be an input wire and output wire? And I like to think that actually this neuron is dancing. I like to think that this neuron knows how to dance. So for example, right now it's doing what I call Bharatnatyam. And then when it decides one of them is going to become the output wire, the axon, it will then do Kathak. I'm going to grow and grow and grow and grow. Yeah. That's happening every time any circuit formed in our heads. And it's happening in a coordinated fashion where the neurons know which way to grow. Okay. What I'll talk about today is then, here is a neuron in the cerebral cortex, one single neuron. Here is a column of them, and they are stacked in layers. It's almost crystalline in that each layer actually has a job. I'm going to talk about the firstborn cells of the cortex called the subplate, which have a critical job in deciding how sensory information enters the cortex. The cortex is like the CPU. Sensory information has to enter it. Okay, from the skin, from the eye, from the ears. How the subplate is like a gatekeeper for sensory information. Then there is layer 6 and 5 and 4 and 3 and 2, and I'm going to talk about some of them. Layer 2 and 3 are the ones that connect to the other hemisphere. Okay. Layer 5 and 6 connect outside the cortex. So layer 5 goes down to the spinal cord and controls movement and so on. Layer 6 connects back to the sensory input that it got. And layer 4, which I will allude to, layer 4 is what actually receives the sensory input, but only after the subplate has had a say. Okay, so this is the basic wiring diagram, and I'm going to show you glimpses of data from my lab about some of these, okay, about subplate and about layer 2, 3, and I will allude to 4. All right, here we go. Okay, so that's that connection, okay, is critical. And why is it critical? Sensation has to enter the brain from all over the body, right? Touch from your tip of your toe everywhere. Eyes are placed here, ears are on the side. How does all of this enter cerebral cortex? The roadmap is complicated, right? We cannot have wires going from here direct. So all the sensory information is funneled to a structure at the center of the brain called the thalamus. Okay, the thalamus is not all one big mush. There is the touch thalamus, there is the auditory thalamus, and the optic thalamus. There are different structures. All the information comes to the thalamus, and from there, the information sort of grows into this cortex and fans out. And this map is very reliable. In every little mouse that's running on the street, and in every little human ever born, in every mammal, visual cortex, sensory cortex, okay, motor cortex, premotor, frontal cortex where association happens, learning and memory happens in the middle, there are two hemispheres, and auditory happens somewhere on the side. Okay, this map is very, very reproducible, conserved through evolution, and robust, which means that there are no mutant mice or humans or whatever where visual cortex is suddenly in the front. Because if that were to happen, enough other things would go wrong that the embryo would not reach that stage of forming cortex. Okay. How is this map so reliable? Humans have such big brains. Oh, we're so proud of our big brains. Mice have small brains. Well, all this patterning happens when the embryo is tiny. Okay, although the mouse's cortex, when you spread it out, is like a 1 rupee coin, and the monkey cortex, when you spread it out, is like a uttapam, and the human cortex, when you spread it out, is like an extra large paper dosa. All of them started small, and when they started small, that's when gradients of signaling molecules set up the map so that you have this reliable patterning. Okay? All of the sensory information comes into the thalamus. From there, optic information will go to the visual cortex, auditory information will go to the auditory cortex, sensory information will go to the sensory cortex. This is called the thalamus to cortex pathway. In all these cortices, it will first stop at the subplate and then go to layer 4. Why does it stop at the subplate? What is this subplate thing, right? It stops at the subplate because layer 4 isn't born yet. Think of the coordination. So much has to happen, right? So the mother cells of the cortex have barely made the subplate and then made layer 6 and 5. And the thalamus is already ready because the rest of the body has formed. The thalamus is already ready and the thalamocortical pathway is already there. So the subplate can be seen as a park and hold array, wait. So if axons don't get targets, they shrivel up and die. So the subplate is a transient target, wait, tera time aega. And then when layer 4 is born and migrates to the right place, postnatally, post-birth. So this stopping happens by embryonic day 15 of a 19-day gestation in mouse. And layer 4 is finally in place by postnatal day 4 or 5. So this entry happens only after 5 or 6 days of waiting. And if this waiting period doesn't happen, the mouse is sensory blind. Okay, not because there's anything wrong with its eyes or ears or skin, but because the pathway hasn't wired up. Again, wiring of circuitry is everything. So I've essentially summarized the first bit of data. Okay, that is a beautiful cortical neuron, and now we're going to talk about a few of these layer-specific functions. Okay, and then I'll wrap up. Okay, there are 2 genes that I want to introduce because we're going to look at mutations, mice that are mutant in these genes. One of them is LHX2 and the other is FOXG1, and I'm summarizing data from humans. Okay, I don't work on humans, but the human connection of our work is obvious because of these studies. This one by Schmidt et al. LHX2 haploinsufficiency, that means one copy being mutant causes problems. In mice, we have not found haploinsufficiency problems. We have to mutate both. Okay, but for humans, if one copy is not there, the levels, the threshold seems to be such that it causes problems. What problems does it cause? Well, autism spectrum disorder, variable intellectual disability, and microcephaly, small brain. Okay, and this information came from 19 individuals from 18 families, each of them having mutations in LHX2. FOXG1 is better known. There is a syndrome called FOXG1 syndrome. Okay, haploinsufficiency again, and identified, diagnosed when there is irritability, inconsolable crying, loss of language. So it's part of the autism spectrum thing. And the FOXG1 is heartbreaking because there are more than 120 different mutations identified in this gene. So this gene is something of a mutation hotspot. And both these are fundamental transcription factors, and mistakes in them can cause devastating effects in humans. Our work in mice over the years has identified each of these genes having different roles at different times. So LHX2, which we have worked more extensively on, has different roles when the subplate is being born, different roles when layer 2 is being born, a different role when, okay, we have actually parsed out all of these roles and all of them become relevant to the human condition. Okay. So let's begin with the sensory brain. Remember I said sensory information eventually enters the layer 4 in the sensory cortex. Now layer 4 is easily identified in the sensory cortex in the mouse because you see little structures in there called barrels. What barrels? Okay. So in order to go into barrels, I will first talk about how the human brain is actually differentially, how do you say it? The human cortex represents touch differently. Okay, this weird animal, human, is in proportion to how much of its sensory cortex is accessed by, let's say, its thumb and forefinger and mouth and tongue. So basically higher resolution is achieved by devoting disproportionately large territories of cortex. It's like buying a great big, 8 gazillion megapixel camera. Okay, when you want sensory processing from what is relevant, the back of your hand, less sensory resolution. Okay, that's in humans, and there is a mouse equivalent. For mouse, the whiskers are like fingers. The mouse actually whisks through a gap before it goes in, and each whisker is again represented in the brain in accessing much greater territory. So the whisker and the mouth parts again, and look at the forelimbs, the same deal, right? When you look at the mouse's brain, you essentially see where input from one whisker has come in and the cells of the cortex ring around it. Okay? So those are called barrels. So here we are looking at the brain where the whisker innervation has accessed it, and this is high-magged here. So each of these whiskers, input from them goes through the hindbrain, through the thalamus, comes to layer 4, and there cortical neurons ring around it, forming these, and there's one for each whisker. And this is a great accessible system to study Plasticity. If you remove a whisker, for example, that territory in the cortex will be taken up by other neighboring whiskers. Cortex never sits around idle. In blind people, the visual cortex gets taken over by touch. Okay, cortex doesn't sit idle, and this is a great model system for this reason. Okay, are there really barrels in the brain? Well, this name was coined by Woosley and van der Laus in Lausanne, and obviously they might have been inspired by stacks and stacks of beer barrels, and this is how it actually looks. When you take a 1-week-old control mouse and you stain it up with cytochrome oxidase, which is an enzyme that is seen in the tips of the thalamic axons that have come in. Okay, and you can see each barrel. Now, my former student Ashwin Shetty examined a mouse in which LHX2 was mutated in the cortex. We did nothing to the thalamic axons that were coming in. They were completely normal. We mutated in the LHX2 in the cortex, and we couldn't find a barrel. We couldn't find a barrel to save our lives. So what happened? Initially, our hypothesis was, hey, maybe the cortex is hostile. These completely normal axons are coming in, and the cortex maybe won't let them. That's why we don't see a barrel. So then we began. So what is this knockout? This knockout is only in the cortex, in the early embryo, and which cells of the early embryo? In the dividing stem cells that are going to produce the cortical layers. Okay. If you wait for the layers to form and then remove LHX2, you don't get this problem. Okay. So here is the schematic. Here is one neuron in the thalamus, or maybe a cluster. That is the curved thalamus to cortex pathway, and here is how it penetrates the cortex. And here's the actual data. So we do in utero electroporation to introduce a green fluorescent protein encoding piece of DNA into the thalamus. And here, so the connection here is just not in the plane of the section, but you can see the thalamic axons obediently waiting. They haven't yet innervated this blue layer of cortical neurons because this is still embryonic day 15. Suranjana asked, well, let me see whether, okay, so in postnatal life, we know that you don't see barrels. The axons haven't gotten into the cortex. Suranjana asked, well, let me see in the embryo. You know, what do they, is this non-entering the cortex also disrupted early on? And what she found, together with Geeta and Tuli, who did the very difficult technique of in utero electroporation, okay, which is this: you introduce in utero, you open the mother under anesthesia, you put in DNA, green fluorescent plasmid into the ventricle, you apply electric current, the membrane pores open briefly, the plasmid goes in, and that's still the mother cell. So every neuron they produce is now going to be green. Okay? And that's an example. So each little ball is one embryo. This gives you a sense of scale as to how difficult this surgery is that Geeta and Tuli did. And here's what we see. In the normal 1-week-old mouse, you see barrels, now in green. And this is a high mag view. You see this is the pathway that has come in, and these little clumps are the barrels. In the cortical stem cell knockout, you don't see a barrel at all. In fact, you see the pathway that has come in, but it hasn't entered. And Suranjana asked, well, you know, if this is the 1-week animal, what is the picture in the embryo? And here is where we got our biggest shock. This is the control embryo, axons waiting obediently. They haven't entered because layer 4 is not there yet. Here is the mutant. So rather than hostile cortex, which was our hypothesis, we now see an overabundance of axons entering in, and we began wondering, why do these axons jump in? What happened to the subplate? Is the subplate not there? Because remember, the subplate is supposed to be this array. Here's a high mag view of the same thing. Okay, the subplate is a park and hold array for incoming nerves, and this was supposed to happen. Did it not happen? Did the subplate basically not exist because we have removed LHX2? And to our great surprise, what we found is that the subplate was very much present but electrically dead. Okay, this is the collaboration with Upi Bhalla's lab at NCBS and his student Dipanjali Dwivedi. Suranjana went there with mice, took them at the right age, and essentially found that where at the embryonic stage of embryonic 15, control neurons are spontaneously active, or a good fraction of them are. Okay, these blue spikes are all just spontaneous action potentials being fired by control neurons. Mutant neurons, 88% of them are electrically silent, okay, compared with the 36% in control. And if you inject current to force them to fire, okay, a control neuron will fire for the duration of the electric pulse, okay? But a mutant neuron, try as you might for different grades of picoamps of current that you inject in, a mutant neuron will be reluctant to produce even one action potential. So essentially, we found that though the subplate is present, it's electrically dead. And that is why the entire sensory pathway basically doesn't notice the subplate is there, rushes in, but there's no target. And remember what I said happens when axons don't find their target? They wither away and the thalamus dies. And that is why this mouse, which has a perfectly good sensory peripheral system, can neither see nor hear nor feel touch, because the thalamocortical pathway didn't enter or rushed ahead and entered cortex and then withdrew and died. Okay? So this is our study about how sensory input enters the brain and how we have identified LHX2 to play a key role in the subplate's electrical activity. To date, people do not know what it is that the subplate gives to the thalamic axons, and this offers the first insight. Now, I'm not going to spend time on layer 5 and 6 and 4, but I'm going to give you one glimpse of what the same transcription factor LHX2 does. Now in layer 2, 3, a completely different function, okay? It doesn't make them electrically dead or anything. It's a completely different function. Okay, so this was subplate. In layer 2, 3, so the control is down, the mutant is up. In layer 2, 3, I don't even need to tell you what it does. It makes a neuron that looks normally like this into a neuron that looks like this. It shrinks the dendritic arbors. It shrinks the input wires. Okay. This is the work of Mahima Bose, a former PhD student who is now at Stanford. And these are neurolucidar tracings. And here's the quantification. Essentially, the dendrites are shrunken, but not the axons. So these layer 2, 3s are cells that send their axons all the way to the other hemisphere. And look, in the mutant, it seems to be completely normal. It's even penetrated the layers of the opposite cortex. Right? So it's not generically that LHX2 messes up everything. In the subplate, it made the cells electrically silent. Here, we've actually collaborated with Rishi Narayanan's lab at IISc. The cells are electrically very much active. In fact, they are overexcitable because they are small. Small neurons are more easily excited because there is less membrane to bring to threshold. So they are happily excitable and overexcitable. But their dendrites are shrunken. Their axons seem to be perfectly fine. So a completely different phenotype uncovered by removing LHX2 at the right time when layer 2, 3 cells are being born. Okay? So that was the story about layer 2, 3. And I'm going to now close. You can see I have only a few slides left with a superb, unexpected finding that ended up being the second half of Mahima's thesis. Remember, I introduced two transcription factors, LHX2 and FOXG1. Other work in the lab had shown that very early in development, FOXG1 is upstream of LHX2. That if you lose FOXG1, you lose LHX2. This was the work of Geeta Godbole, a former postdoc, who did the in utero electroporations for the other study. Mahima found this dendritic shrinking problem when she removed LHX2. So she said, well, let me ask if FOXG1 is what regulates LHX2 normally. Let me remove FOXG1 and see if I get the same issue. Do I get shrunken dendrites? She removed FOXG1, and I wouldn't be telling you this story if she got a completely different result, and that's what I'm going to close my talk with. Completely unexpected, and that's how her thesis ended up having two completely different halves. Okay, one gene is supposed to regulate the other, but it does so at stage A, but obviously not at stage B. Otherwise, you wouldn't have got this problem. What did she get? Here is a typical cortical neuron. They're called pyramidal neurons because of their shape. And these red cells are the support cells around them, the astrocytes, the glia, star-shaped astral astrocytes. Amazingly, both these come from the same mother cell. So in the central nervous system of vertebrates, the same mother cell first produces neurons, and then when it's finished producing neurons, produces glia. It makes sense that you first make the circuitry and then you put in the support cells. These support cells do more than just provide nutrition and so on to the brain, to the neurons. They also mediate neuron to neuron communication. Okay. What Mahima found, together with Varun and Urvi and Ishita, what she did is she removed FOXG1 by electroporating at embryonic day 15 when the upper layer cells were being born. Remember dendritic problems and so on? That's what she was looking for, and looking at postnatal day 14. So you harvest the brain 2.5 weeks later. And what she got, okay, this is the control. At postnatal day 14, these are layer 2, 3 upper layer neurons, and you can see that they are actually new N positive neurons. They did not have dendritic shrinkage problems and so on. But you remove FOXG1 and there was not a neuron to be found. What she did was she essentially converted all neurons into glia. The progenitors simply stopped making neurons. They prematurely began making glia. Okay, this is a glial marker. All these lovely bushy cells were all positive. In fact, this is the quantitation. Okay, for the neuronal marker nu n, that's the control. We couldn't find a single neuron to save our lives. Okay, and they were all hugely increased in the glial marker. So she essentially prematurely induced the neuron to glia cell fate switch. Completely different finding and unreported in the FOXG1 literature. So this is her contribution to the entire FOXG1 autism spectrum possible things that could underlie the ASD. Then COVID happened. Okay, Mahima and Varun, undaunted, they were the only two people who stayed back in the lab during COVID, and they basically found how FOXG1 does this by bioinformatics. They narrowed down candidates by doing bioinformatics in COVID, and that's close to my last slide. Eventually, we did an RNA-seq, okay, control and mutant, and we found genes that went up and genes that went down. But they first looked at a FOXG1 ChIP-seq data set to identify which genes does FOXG1 sit on, potential targets. Then, of these, which genes have bivalent chromatin marks in neural stem cells. What's bivalent? It's like putting your foot on the brake and the accelerator at the same time, so that you release one or you release the other, you're poised to either stop or go. Okay? So chromatin, DNA complex with proteins, can be marked for either upregulation or downregulation. And she asked which of these genes are also marked bivalently in neural stem cells. And then and doing that, which of them are enriched in astrocytes, and that gave a list of about 19. And then when they overlapped that with what genes were upregulated in the RNA-seq, they found FGF receptor 3, which was fantastic because the FGF pathway is a strong progliogenic, known progliogenic pathway. So essentially, to summarize, Mahima found removing FOXG1 from a mother cell upregulates FGF receptor and makes that cell more sensitive to FGF signals. And since FGF is gliogenic, that's how that cell now produces a glia instead of a neuron. And she in fact showed this by using an FGF inhibitor, which I'm not going to show you the data, but it's published, that using an FGF inhibitor and this process gets stamped. Okay, so she essentially found two completely different sort of mutant phenotypes of two completely different transcription factors. Yeah, so I'm going to just summarize here. But what was going to be one story ended up being two stories for Mahima's thesis, building upon the work of Suranjana with the subplate. So what we are trying to do is essentially find how each of these layers and their connectivities are shaped, so that we can ask how does this fantastic cerebral cortex get wired, so that it can enable the functions that we need for survival. Image completion, matching auditory and visual signals, the ba-ba-ba-ba experiment. And eventually, maybe, we will sort out how we see illusory contours that allow us to live. With that, I will thank the funding agencies that supported our work. And you have such a beautiful campus, so I wanted to show off we also have something nice. And I'm happy to take questions.
Thanks for the wonderful talk.
Thank you, ma'am, for the lovely talk. I had this question, like you said, like other than brain, other organs like are transplanted, but they also have their own circuitry. Once you remove the organ, you cut the nerves and everything. So how do they rewire that?
Lovely question, lovely question. Thank God and nature and biology and evolution that the peripheral nervous system has no problems regenerating. Okay, so you cut and you sever a nerve, the peripheral nerve will regrow. You will be deadened for a while and then you will feel tingling as it regrows. Okay, central nervous system does not regrow. We can hand wave and say, oh, because those central circuits, once they are made, how can you regrow them? They'll get messed up and so on. The key problem there is the central nervous system myelin. It actually has a protein that's been named no-go. So, if only spinal cord victims who have spinal cord damage and accidents and so on, if only nerves could be persuaded to grow past the scar tissue and so on, but central myelin does not allow that. Peripheral, thank God, does. So your problem is solved.
Thank you, ma'am.
Thank you, Professor. It was a brilliant talk. I had two rather general questions. So apologies at the outset if they are a bit silly. I'm not very familiar with the area as such. In the beginning, you described about how there are these input wires and one is chosen to become the output, right? So I was interested in knowing how that choice happens, and in vitro, have they shown through any electrical or chemical bias a reproduction of such a phenomenon?
So the final answer on that, so if you look up the work of Gary Banker, that lab began with trying to ask, is it length? So they spent time trying to cut the longest wire, asking, does it switch? The answer is that axons have microtubules oriented all in a uniform direction and dendrites have them mixed. And the centriole plays a role in this process. And I don't actually know how the debate sorted out, but if you look up the centriole and microtubule orientation literature, that's where the answer is. I don't actually remember the.
Right. And later when we talked about the layers, right, after the subplate forms, we have layer 4, which, if I understood correctly, collects the inputs, and then the outputs come from 5, 6, and the rest respectively.
Yes, exactly.
Why isn't the input layer closer to the subplate? Why do I have 5 and 6 and then 4?
Okay. In biology, we do not ask why. Abhi kya kare, there is a better, logical, cleaner way of building almost any biological thing, but living systems are constrained by evolution, and you would ask, so layer 2, 3 are more expanded in mammals that have more cortex, or in vertebrates that have, so neocortex with this particular layering is available only in mammals, but even reptiles need to move. So layer 6, corticospinal tract, is required even in reptiles. So you could ask there also, is the output more important or is the input more important? Somebody has to be closer to, right? So layer 5, 6 happen to be that way, but there is no logical answer. You could construct the brain completely differently and have it work, but evolution did not allow for that.
Thank you, Rosa. Again, brilliant talk. Thank you very much.
Any other questions?
Shubha, about the LHX2 function in the subplate, is it leading to a reduction in the electrical activity, or is it possible that this input neurons are making aberrant connections? They enter early somehow due to an attractive factor, and that would lead to an electrical problem there. Is it possible?
Are the other neurons becoming somehow more attractive?
Correct. And then
so the subplate is no longer an attractive target. Excellent, my God. So luckily, Suranjana did a beautiful series of experiments using Cree-ER, where she was able to mutate LHX2 not just at day 11.5 and subsequent, but she gave tamoxifen at day 12 or day 13 or day 14. So she found that she got this phenotype only when she gave TAM at E11. Only when the subplate was being born. Okay. If she allowed the subplate to be normal and then gave TAM at day 12, she didn't see this problem. Okay. Wow. So we narrowed it down to the progenitors of the subplate, but we had to do that experiment. So you really...
Sure, thank you.
Yeah.
Thank you.
From chemistry point of view, When you speak of proteins, yeah, proteins are made up of enzymes or proteins, and proteins are made up of amino acids. Speak of cell membrane, yeah, lipid bilayer. How do you describe what is the chemical composition of a neuron?
Chemical composition of a neuron.
Yeah, what actually is it made up of?
What every cell is made up of. Lipid membrane, all of the organelles.
It has everything?
It has everything other neurons have, the other cells have. Neurons are special only.
So can you call this as a cell by itself?
A neuron is a perfect cell. It's just a specialized cell that is electrically active. So it has ion channels in it that respond to changes of, that open and allow ion flux. And when it reaches a particular threshold, more ion channels will open up. So you get what is called an action potential. Sudden change in depolarization.
In other words, all constituents of this cell
Everything.
are present in this?
Everything, absolutely everything.
Absolutely?
Absolutely. So neurons, because they're electrically active, invertebrates are myelinated, they're insulated.
So what actually in it basically kind of makes it respond to electrically, right? The composition has
Why
to be
is a
different.
neuron electrically active?
Absolutely, yeah.
Actually, this was a question on my comprehensive when I registered for PhD. Seriously, Henry Lester, king of ion
So
channels.
I'm not appearing for any.
It was fantastic. I mean, it was, how do you make a cell? So he said, take a lipid membrane. It's a ball. Now it's a cell. Now endow this cell with the ability to, to pass electric current. What channels would you put in? Now make this electrically active cell where it can regenerate its, where it can build upon its depolarization. So which channels will give which property? So a neuron is just a collection of the appropriate type of channels with the appropriate gates. Some of them are voltage gated, some of them are, so the initial channels that open are chemically gated. They respond to neurotransmitters. When the neurotransmitters sit on the channel will open. But then when the voltage drops to a certain threshold, voltage gated channels will open. And that's what makes the action potential fire. So it's actually, you might say it has now gone from chemistry to physics.
Okay, too complicated.
So very exciting talk. So couple of questions, only two parts. So what we are talking is mostly intrinsic network inbuilt, but how much of it depends on the external stimuli when it develops.
Excellent.
And following up with the age, how it changes.
Excellent. So circuit band to gaya, but can it be shaped by experience, and what happens when it gets damaged, right? The intrinsic ability to, say, reach layer 4 is programmed, but how inputs from the two eyes get integrated in layer 4 so that you can have binocular vision and see depth, that requires visual experience. So if one eye is closed in a critical period in early development, the inputs from the other eye will take over layer 4 and this eye will not. So the actual wiring has to be like alternate. Yeah. So this was a Nobel Prize of 76, Hubel and Wiesel, that they found ocular dominance columns and how experience was required. So that's some aspects of functional circuitry require experience. You have seen a newborn, right? They lie there kind of helpless. Newborn humans. Newborn calves will get up and run because their circuitry is wired up. But newborn humans will lie there, and periodically when they are like 2 months old, they will do that. Why? Everything is wired up messily. So the nerves that simulate the biceps and the triceps are all mixed up. So everything contracts at once. The circuitry has not learned that some neurons will only go here. So this is called synapse elimination, where if they are connecting two opposite muscles, one has to go. That requires motor experience. So the small movements that happen in utero onwards shape that. So that's normal development. What happens in disorders? Absolutely, circuitry can. So multiple diseases of myelination. If the myelination goes awry, then the nerves don't function, then the muscle will not function. So there are many ways these things can go wrong. But we need to understand how it is normally built in order to then look at disorders. So thanks. You actually presaged a completely different talk.
Ma'am, here.
See, sound localization. I look at the.
It's a really wonderful talk, first of all. I don't think like what I will ask, it will be relevant for this talk, but what I can see is like, I can see the regulatory, like the circuits of the brain, like kind of a Boolean network, like on-off switch kind of thing. What I what I know is that the cancer gene regulatory network is very highly studied using Boolean networks, making all that patterns and connections. So can we study like the brain circuitry by using Boolean networks, by like each neuron as a node, and every node is an on-off switch, and we can come up with such equations and all?
Absolutely. So the work I know is that of John Hopfield, so you can look it up, Hopfield. He was a professor when I began graduate school, modeling networks as nodes and asking how you can weight them and so on. That is an entire line of scientific inquiry. Exactly. I know very little about it, but you're absolutely on the right track.
Also, like another thing about memories,
memories.
So memory, is it like a particular state of a pattern that is formed in a brain? Like if we recursively activate that state of pattern, it gets stored like a memory in the brain. So is it something like that?
Two things. So what is the neuronal physiological correlate of memory is what you're asking. Look up the terms long-term potentiation, where a weak input might normally stimulate a neuron to a certain extent, but if that weak input is paired with a very strong input, as in something memorable, then the response to the weak input will then be much stronger than it would otherwise have been if the strong input hadn't come in. Long-term potentiation. Memory for specific, so this is best studied perhaps in the odor space, where how does a fly or a more tractable animal learn to distinguish between 10 different odors. So you can imagine odors being encoded in a network as zeros and ones, and each odor, so one neuron cannot only participate in one odor, because otherwise you cannot have one neuron dedicated to remembering, say, your grandmother. Otherwise, if that neuron goes, there goes your grandma. So memories are stored as strengthenings of patterns of synapses between multiple neurons. And this pattern can be corroded to a certain extent, but you will still retrieve the memory. If it corrodes enough that it overlaps with another memory, you will actually remember a different memory, and you will be sure you have remembered that.
So it's basically the strength of the neural network.
Strengths of synapses and long-term potentiation are the two keywords I can give you to read more.
I will check this out.
Yeah.
Thank you.
Great question.
Good evening, ma'am. I had two questions regarding your talk, one of which was how do the thalamic axons find and connect with the subplate during development? And the other was what happens to the subplate neurons once the cortical circuits are permanently formed?
Okay, brilliant questions. The second question is easier. The subplate is a transient structure. So once it's done its job, I don't know why these extremely glaring lights are put on the speaker, so I will talk to you like this. The subplate is transient. It stays long enough to help the circuitry to form. Now, the first question, how do the thalamic axons find the subplate? Fantastic question. I devoted 4 weeks of my advanced course to this question because the thalamocortical pathway undergoes such a convoluted route. There are intrinsic mechanisms in the thalamic neurons and guidance as it takes a U through the base of the brain. In that U, it splays out according to visual and auditory and so on. Then it enters the cortex such that the axons are in the right place. And then there are mechanisms in the cortex. So there are multiple stages of mechanisms. And this is really the topic of an advanced course. But a review on this. Okay, my student was working on it. We stumbled into this. We didn't expect our LHX2 mutant to have a thalamocortex. We weren't experts in this, so we stumbled into it. My student, Vanisha Lakina, finds this review. The first sentence of which goes, whoever works on the thalamocortical pathway is either a sadist or a masochist. And she goes, Shubha, you're the advisor, I'm the student, so you're the sadist and the masochist. Okay, so very complex to sort out.
So do the axons spread out once they cross the U and then?
The cortex is very big. They are already spread out.
Okay.
I show it to you in a section, so you think it's one tight thing. But if you see, right now, if you're looking this way, you see one tight curve. But if I turn it this way, you will see a spread.
Thank you so much, ma'am. It's a
Three-dimensional.
brilliant talk. Thank you so much.
Thank you for your inspiring work and share your knowledge about brain development, ma'am. And I have a question that since the human brain has about 100 billion neurons, so if a person, hypothetically, if a person had more than 100 billion neurons, would that increase the number of neural circuits and thereby enhance cognitive ability and memory?
Okay. You must be a computer science major. Having more cores, having more is better. Okay. Having a neuron doesn't mean it's going to wire up properly. So everything is in the dendrites, right? So having the average number of neurons. Okay, I'll give you a nice hot potato example. The brains of women are 20% smaller than the brains of men. You want to take that one on? Fewer neurons.
But Marie Curie...
It is how it wired up. Okay, it's how it's wired up. So body has to be accessing the sensory cortex. So a whole lot of the area is sensory cortex. How much cortex you devote to what? Within that, how the neurons are expanded and wired. One neuron participates in so many functions that how it is wired up and how that wiring is trained during early experience is key, not how many neurons you have. And remember neuron glia cell-fate switch? So suppose we operated it the other way and didn't make glia and just made more neurons. Those neurons wouldn't work because when two neurons talk to each other, there has to be an astrocyte monitoring that. And when one dumps glutamate onto the other, what happens to the extra glutamate? The astrocyte takes it up, makes into glutamine, then pops it back out. So unless you have all of that, more is not always better. It has to actually be wired up properly.
So means I have a doubt that if more neurons, more cognitive ability, yes or no? Means that's the thing.
More cortical territory, more resolution. So depending on how much you devote to a particular modality, that modality is going to have more processing power, right? So blind people, congenitally blind people, have exquisite touch. They have devoted much more of their cortex to it. So yes, yeah. So anything that you do that trains a circuit will make it grow. So yes.
One second question, last final question.
Hello, ma'am. Thank you for the lovely talk. My question is regarding synesthesia. So, as we know that in synesthesia, synaptic pruning happens. My question is, how early does this pruning happen? And if there are developmental cues which will dictate whether adult brain will be a synesthesious brain or a normal brain?
Okay, I have no idea.
Okay.
Because to date, nobody will give me their little child to put this gene or that gene into and, or be able to predict whether the child will be a synesthete. So I have no idea. It's very difficult to answer that question because, more seriously, synesthesia is not a uniform trait. Different synesthetes have it to different extents. So for people who don't know what it is sometimes numbers will evoke the sensation of color. And possibly because there are cross pathways between the color coding pathway and the numerical processing pathway. But different synesthetes have different extents and different versions of these issues. So since you can't repeat an N, and you can't do experiments on humans anyways, and since no mouse will tell you whether it sees numbers as colors, setting up the problem tractably is what's the challenge. Yeah.
Okay, one last question.
Hi, Shubha. Excellent talk. One very technical small question. On the subplate neurons, you mentioned that they are not firing anymore, therefore they are bypassed. If we forcefully make them fire, can the phenotype be recovered? And the loss of firing is due to some loss of ion channels here or something else?
All unexplored questions. I never actually showed the therefore. I showed loss of fire. The neurons are silent and they are bypassed. We were unable to make the connection because we are unable in terms of reagents. into rescuing the electrical activity in vivo, and so on and so on. We're unable to do it technically. If that were rescued and then the neurons grew, we would then be able to say therefore, and then we would have found what the subplate gives to the thalamocortical pathway. So, are there ion channels changed? We did an RNA-seq. We have a whole bunch of changes, including ion channels. Yeah.
All right, so I think in the interest of time, we have to close the question and answer session. I'm sure there are hundreds of other questions which will have to be taken offline. But let's all thank Professor Shubha Tole for this wonderful talk. And I would request our director, Professor Jain Murthy, to hand over a small memento to Professor Tole as a token of our appreciation.
Now, the real thing is, will I
wonderful.
actually get the memento? Because this was a test. Would the talk be accessible enough was the test. So if he gives it to me, it means I've passed.
Well, the questions make sure it is accessible.
It still bothers me, the way that I've learned the cell is different, but then you see all kinds of lines there. I was just bothered by that. What actually are the lines made up of? Like the dendrils that you got?
Membrane with microtubules.
Yeah. So, the cell that we learn is different, right?
That's true.
So what you are showing is different.
That's true.
Sorry for my limited
Milega ki nahi, milega ki nahi.
Yeah, wonderful.
Thank you.
For the institution.
How beautiful.
It's
Really beautiful.
Really.
Yeah.
Thank you. Thank you for the nice talk. Yeah, I think our seminar speaker, yeah, please go ahead. So,
That's just an extra cup for you.
Thank you. Wonderful.
Right. So thank you, everyone, for coming here and making this talk a wonderful occasion. So, Tole will be here for a few more minutes, so I'm sure students would like to mob her and ask more questions.